Okay. So we know everything there is to know about red blood cells. What's another name for a red blood cell? Erythrocytes. And what are we talking about here? Leukocytes, which is a fancy name for what? White blood cells. How many different types? 5. What are they? Neutrophils, lymphocytes. Never let monkeys eat bananas. Neutrophils, lymphocytes, monocytes, eosinophils, basophils. Why did I put them in that order? Yeah, so the most numerous is the neutrophils. That's why, remember the little never let, and it's actually in the book, never let monkeys eat bananas. Okay, neutrophils, lymphocytes is next in line. Then we have the monocytes, the eosinophils, and the basophils. Okay? 2 divisions of these guys, leukocytes. Remember, a leukocyte, that's all of them. Lymphocyte, that's one of them. Those 2 names are pretty close, so be careful. So we can have granulocytes. And what did I say about those last class? Why do we call them granulocytes? They have little granules in them visually. They are the eosinophil, basophil, and neutrophil. And then we have the agranulocytes, which are the monocytes and the lymphocytes. Now, different jobs, different functions these guys have, and they're going to be stimulated by different things. What stimulation did I get when I needed to make more red blood cells? It was a hormone, right? Made by who? The kidneys. And what was it called? Erythropoietin. Whatever. Now, we have a bunch of different things that can stimulate the production of white blood cells. So depending on what's going on in your body, I might need more of one of these guys to do a job. We see, and let me see where it's described in the book. Okay, on page 646, production and lifespan of leukocytes. There's a little blurb on some of the different chemicals that will cause the maturation of the hematopoietic stem cell, or the, what's another name for it? Hemocytoblast. Remember, that's the guy that lives where? In the bone marrow, red bone marrow, where all blood cells come from. But the stimulation to make one of these guys, one of the 5, is going to be different. Terms I want you to remember: interleukins. And again, these are different substances the body produces under different circumstances to stimulate the maturation of that cell. And colony stimulating factors. So different substances for different cells. So they give us an example. Colony stimulating factor G is going to cause the granulocytic series to start. So instead of hemocytoblast turning into a lymphoid stem cell, it's going to turn into a myeloid stem cell. From there, it's going to get other stimuli to become one of the 4 it can possibly become. But I'm not going to get crazy about which one does what. There are very specific chemicals that will cause the maturation series to progress into one of those 4 guys. Does that make sense? So different chemicals, different maturation series. So remember interleukins and colony stimulating factors. Okay? And I'm good. So what do each of them do for a living? Well, the neutrophils, the most numerous, are phagocytic cells. And these guys are all over the place because they are the most numerous. And I just want to point out one thing. God, I hate computers. You're going to do this to me now, aren't you? Oh, crap. Oh, there we go. All right, see this chart? One of the problems with this textbook is they're going to give you normal values. When we talk about lymphocytes, monocytes, all those guys, the leukocytes, you are going to see normal numbers that are different throughout the book. They're different here. If you look on the chart that's over on page 645, they're different. And in the lab book, they're different. Which one do I want you to know? It's on page 644. So when you do your white blood cell differential count, you are actually going to count 100 white blood cells. And how many, if you're normal, should you get of the neutrophils? Of the 100, about 50 to 70 of them, or because I'm counting 100, yes? Now you see why I count 100? Huh? 50 to 70 of the cells you count should be what? Neutrophils. So when you do your differential count, most of the cells you see are going to be neutrophils. Eosinophils, 2 to 4. You might not even see one in your normal blood smear. Basophils, can you have a half a cell? No. So 1. And again, you might not even see it. Lymphocytes, how many? 25 to 45 of the 100 cells that you count will be lymphocytes in a normal white blood cell differential count. And how about monocytes? 3 to 8. So most of what you're going to count when you do your white blood cell differential is going to be neutrophils and lymphocytes. Yes? Well, that's a range. The number you get is only going to add up to 100. Okay? Make sense? Okay, that's what I want you to know. So, neutrophils are phagocytic cells. We already talked about its other name. What was its other name? Polymorphonuclear Leukocyte. Okay, and everybody understands what that word means. Nucleus of many shapes in a white blood cell. So they have multinucleated, they're not multinucleated, but multilobuled nuclei. So the nuclei of this cell is here. And it's in different lobes. See it? The background. What does the background look like? Grains of sand. And I describe them as pinky purple grains of sand, because they're picking up the neutral portions of the dye that I'm going to be using, which has a whole bunch of stuff in it. It's called right stain. And the granules contain, they're actually little vacuoles that contain chemicals. What do you think the chemicals are for? Killing stuff. Because what does a neutrophil do for a living? It phagocytizes foreign particles. So remember when we talked about phagocytosis? Way back when, more of that stuff you were supposed to remember. So I bring in a particle, nasty bacteria. And then what do? I fuse the particle with a bag of lysing chemicals. So I blow it up, yes? And then what do I do to all the little dead bacteria parts? Throw it out to trash, exocytosis. Does that ring a bell? Okay, this is a phagocyte, so that's what it does for a living. And all those little granules are bags of lysing chemicals. And when I stain it with my differential stain, we're going to see them. And that's the color they're going to be. You with me? So I'm going to see increases, and this differential cell count can tell my physician quite a bit. I'm going to see increases in these cells when there might be foreign particles around, like maybe a bacterial infection. Yes? So my number, instead of being 50 to 70, might be 80 to 90. That's an indicator that something's going on with my patient, right? Neutrophils. Who's this guy? That's an eosinophil. The reason I need to stain it is because I can't tell the difference between a neutrophil and an eosinophil unless I stain it. The little granules here are actually bigger and they're orange. You see them? The nucleus is here, also a weird shape, but the granules are orange. They actually pick up the eosin portion of the right stain. That's why they're called eosinophils. And we see these increase in things like allergic reactions or parasitic reactions, because these guys are going to help with that. Well, if you eat something, you kill it, yes? You can eat live things, right? Sounds disgusting, but that's what the cell does. Once it brings it in, it's going to throw chemicals on it or do whatever. Well, not one. Tapeworm's very, very big, yes? Yes. So in order to fight that off, I must make more, yes? You getting the point now? So eosinophils, they look different. Orange granules, because I have different chemicals in those little bags of mine. Different enzymes that pick up the eosin portion of the stain. Who's this guy? Basophil. And the little black granules, or very, very dark blue, they look black to me. I don't know about you. They do look purple in here, but when you look at them under the microscope, they look black. Believe me. Those granules, again, are bigger, and they're going to pull out the basophilic portion of my stain, and that's why they're that color. And they contain histamines. What's a histamine? It's an inflammatory chemical. It's kind of a signal to all the other guys in a response, an immune response, that they need to come here. And yeah, when you have too many histamines produced in your body, it can cause problems, can it? Yeah. Okay, like? Yeah, like my bronchioles and my respiratory portion of the tubes start constricting, which is not good. Not good. So what do you take? Antihistamines to kind of neutralize the effect of these chemicals. So I want to point out that and you look on your chart on page 647, basophils, and it doesn't show it here, but remember when we talked about connective tissue? We saw some white blood cells in there, and one of them was called a mast cell. In tissue, and these guys can actually do what? They can dance if they want. They can squeeze out of the circulatory system into tissue and do their job too. And in this case, mast cells in connective tissue were actually basophils that squeezed out of the circulatory system to create that response, the histamine inflammatory response that we saw when we were talking about tissue repair. There's another guy we're going to talk about that squeezes out too in a minute. Diapedesis. I think it says it under, does it say it? I know I saw it somewhere. Diapedesis. And they also, under neutrophils, they describe the chemicals it actually makes, and one of the groups of chemicals it makes are called defensins. And we're going to talk about these a little bit more when we talk about immune responses. So the agranulocytes, and we're going to see these guys in several different forms, which is the confusing part of the white blood cell differential. Agranulocytes, this is one called a lymphocyte. Now, we can have small lymphocytes or we can have large lymphocytes. But the characteristics that you should remember when you look under the microscope are as follows. A, nucleus looks different, doesn't it? It's more rounded. It's also much more compact and tight. So it's going to stain up a much darker, thicker purple. The cytoplasm is blue. You see blue? No. You don't see blue? Everybody else sees blue, so get your eyes checked. Blue. Robin's egg. You see purple? It's bluish. Under the microscope, when you look at it under the microscope, it'll be bluish. And sometimes we call it robin's egg blue. But that's one of the characteristics of a lymphocyte. And again, when you go to lab, you're going to see small lymphocytes and large lymphocytes. But they're not going to have that weird shaped nucleus. It's going to be more round and very densely packed. And the cytoplasm is going to be bluish. Yes? What are lymphocytes for? No bacterias. Yeah, they're the viral guys. So if I have something going on virally, what do you think I might see in my patient's white blood cell differential? An increase in lymphocytes. Yes? Okay. Again, when we talk about the immune system, we're going to talk about their specific role in an antigen antibody production and reaction. But they are going to increase in number. Some of them are going to go help the immune response, and some of them are actually going to produce something called a plasma cell. And that's the guy that's going to squeeze out into the tissue, and we see that on our chart, page 647, and become something called a plasma cell. Plasma cells, does anybody know what they do for a living? Because you studied the chapter before you took your quiz? They make antibodies. Okay? We're going to see when we do immune system that the lymphocyte can come in two different forms. One can be a T cell, one can be a B cell. And the B cells are the ones that can differentiate into plasma cells. Those are the guys that make antibodies. The T cell, that's kind of the cruise director of the immune response, which we'll talk about later. Can I tell the difference between a B cell and a T cell here? Not really, no. So, small or large lymphocytes, those are some of the characteristics we look for. Monocytes are also phagocytic, but where are they phagocytic? In the tissues. They can squeeze out of the circulatory system too and become macrophages. They can also become something else. Osteoclasts. Yes? Remember that? And what are they going to help do? You've studied this in the hormone thing. They're going to help break down the bone matrix to increase what? Blood calcium levels. Which hormone is going to stimulate them to do that? Parathyroid hormone. Yes? That's the monocyte. The monocyte is, and this is the hardest thing when you first start doing a white blood cell differential, is to tell the difference between a monocyte and a lymphocyte. The monocyte has a much lighter looking cytoplasm. It's kind of on the grayish side. Again, this looks blue, but under the microscope it's almost gray. The other thing, that nucleus, is it really dense and packed and very purple? It's very purple, but doesn't it look like it has white holes in it? Okay, so the nucleus is going to be, and this is the largest of the leukocytes, by the way. So the monocyte is going to be bigger, And it's going to have a much airier looking cytoplasm and nucleus. And usually the nucleus isn't tight and round. It kind of gets weird, kind of like a horseshoe shaped thing going on. But it's not as densely packed. Okay? So we're going to work through a white blood cell differential count in class on, or next week in lab. Again, know the progression. Granulocytes, agranulocytes. Lymphocytes can squeeze out, and this is the squeeze out, guys. Plasma cells. Monocytes can squeeze out and become macrophages, and we're missing one here. What can he become? Mast cells. So add that to the chart. Basophils are mast cells in the tissue, connective tissue. So that is the leukocytes. Disorders of the leukocytes are what? Leukemias. Now there's a whole bunch of different leukemias, right? Depending on which white blood cell is affected. What is the treatment, or what is the best treatment for someone who has leukemias that are out of control? Bone marrow transplant. So what they do is they wipe out your cells because there's something wrong with them. There's some signal that's causing them to create abnormal white blood cells. And what do? They get somebody else's normal cells and they put them into you. They know where to go. They're very, very smart. So they'll go hang out in the bone marrow where they're supposed to, and they'll start to make more of themselves. And eventually they'll start doing what? What they're supposed to do, make normal white blood cells. It depends on the person. But it takes a while. It's going to take at least a good 3 weeks to a month before they're up to snuff. So what do you have to do after you get a bone marrow transplant? No touch any nasty things, right? You have to be very careful of what you come in contact with until your cells kick in, those new cells kick in and start doing their job. Make enough of themselves. Okay? So they talk about leukemias here. Another one they talk about is infectious mononucleosis. What's mononucleosis? It's a nasty little what? Virus. So it is a pain. Now I'll tell you, how would the white blood cell differential count help me? That's the test they do to see if you have mononucleosis. Did you know that? What cell type would you have an increase in if you had mononucleosis? No monocyte, no. The lymphocytes, exactly. And what happens is the lymphocytes kind of start getting weird shaped. They increase in number, and we call them atypical lymphocytes because they don't look like they normally do. They get a little bit bigger and they get a little bit weird shaped. So that's a really quick way to tell if somebody has mononucleosis. You can also test their plasma to see if they have what? If that's a virus, it's a foreign substance. What am I going to do? I'm going to create an immune response to that virus, and I'm going to create a group of cells that will make very specific what? Antibodies against that foreign substance or antigen. So they can also test to see if you have antibodies in your plasma, and they always do a white blood cell differential. It's just quick and easy. Yeah, it causes problems. It can cause problems with your liver as well. And because it's a virus, and viruses are tricky. We'll talk about viruses when we talk about immune response as well. The next group of cells that can possibly come from a hematopoietic stem cell or hemocytoblast is fragments of cells by the time it gets to the circulatory system. And they're called platelets. Another name for platelets is thrombocytes. What's thrombo? Thrombus. Clot. So what are the platelets going to help me with? Clotting. Okay? They are stimulated by a specific hormone produced by the liver. Guess what it's called? Thrombopoietin. So when I need more platelets, I increase production and release of thrombopoietin made by the liver, and I'm going to cause the hemocytoblasts to mature. Do you have to know the maturation series? Yes, you do. Before I get spit out into the circulatory system, the cell just before that is called a megakaryocyte. Megakaryocyte. And when I go to squeeze into the circulatory system, guess what happens to the megakaryocyte? It gets squished all up and breaks apart. So that's why we get fragments of this cell in our circulatory system. And those fragments are platelets or thrombocytes. And they are very important in hemostasis. The heck is hemostasis? Hemostasis. Stop blood, right? And why do I need to know anything about hemostasis? Oh, look, steps. I love essay questions with nice steps. Steps. Hint, hint on your exam. So, you cut yourself. The platelets are going to help stop that. First thing is vascular spasm. Contractions that are going to cause vaso what? Why do I need to constrict my vasos? Yeah, it's trying to stop you from leaking all over the place. Because of the tissue damage and the release of the chemicals from those cells, we're going to start to attract more platelets to the area of the damage. And they are going to, in step 2, form what we call a platelet plug. So they're going to kind of plug up the hole, like a cork almost. They're going to cause collagen fibers to come to the area and adhere to the platelets and act like a Band-Aid almost that sticks to that damaged area. They're also going to release chemicals. And those chemicals are going to set a whole process in motion that we're going to talk about in a minute. They'll eventually form a platelet plug. The next thing that happens is this chemical event. And it's going to take some naturally occurring, floating around in liquid form proteins and cause them to become solid. That protein is called prothrombin, and eventually it's going to become fibrin when it's in its solid form. So we see in step 3 coagulation occurring. And this is when the platelets, through this series of chemical events, will cause fibrin formation that's kind of adds some mesh to the platelet plug. So now we have something that kind of looks like a scab almost on the inside of blood vessels. In medical lab school, I had to know all of this. And this is what we call the clotting cascade. I won't make you know all of this. I know I'm an angel. Now, you see a bunch of Roman numerals in there, yes? You should learn the Roman numerals because Super Bowl is always written in Roman numerals. And that's important. What Super Bowl is this? It's 46. X, L. L is 50. The X before the L. Take away 10. V is 5. All right. So, Roman numerals, just a quick little, so you know which Super Bowl and you don't look silly when you're asked which one it is. These are what we call clotting factors. And they go through a series, kind of like a domino effect of chemistry, to form fibrin. There's two ways to start this clotting cascade going, and that you do need to know. We can have something called an intrinsic pathway, and that's when things inside the body happen, like blood vessels wear away. And you do this all the time. I mean, there's constantly blood moving through these blood vessels. It's kind of like in Maine, you know, you have to do road repair, right? So you're constantly doing road repair too in your blood vessels. So vessel endothelium, and that's the cells that line blood vessels, might rupture, expose underlying tissue. Stuff inside will set that portion of the clotting cascade off. Or something from the outside, extrinsic. Somebody comes up and punches you and breaks some of your blood vessels. Or you cut your finger off when you're cooking dinner. Yes? Something from the outside will set this off by actually releasing chemicals from those damaged cells, tissue factors. So you have to know the difference between intrinsic and extrinsic. And hopefully it's self-explanatory, yes? Something on the inside is intrinsic and something on the outside is extrinsic. So I'm not going to make you know all the stuff in between, but they both are going to lead to the formation of this. And this is where you have to start knowing this. Where am I going to find this diagram? Page 650, figure 17.14. Now, some very important substances in this reaction, calcium. All of the clotting factors, and if you want to know what they are, there's a table, 17.3, on page 651. There is how many of them? 13 different clotting factors, all important for blood clotting. Does anybody know what hemophilia is? The inability to form a proper what? Clot. Why? One of these factors is missing. Yes? I'm not going to make you know which one. So there are all the factors.
We don't need to know those?
No, you don't need to know those. If you go to medical lab school, you need to know those, but you don't need to know those. The chart where? 651, yeah, you don't need to know. Like I say, there's a bunch of them. Who makes most of them? Liver. When we have patients with cirrhosis of the liver, what's one of the problems they have? Clotting. Bruising. Yes? Why? Yeah, because their liver isn't working well and not making probably the amount that they need of many of these clotting factors. This is what we need to know. So, for whatever reason, intrinsic or extrinsic, I start the cascade and form prothrombin activator. I'm then going to start a series of chemistry that will then take something that's floating around called prothrombin 2 and switch it or change it into thrombin. When thrombin's on the scene, it's going to take fibrinogen, which is hanging around in the circulatory system as a plasma protein, and it's going to cause it to become a solid, fibrin. Yes? And with the help of those last factor 13s, we're going to cause the mesh to form. Again, calcium is involved in that as well. So this series of chemical events happens, and again, we refer to this series of chemical events as the clotting cascade. If I am missing anybody anywhere in this whole event, I'm not going to be able to do what? Form a proper clot. Okay? So know this. Yes? And know how to get there both ways. Either intrinsic or extrinsic. Yes? What's an anticoagulant? It's going to disrupt this clotting cascade and stop this from happening. Because sometimes our patients can have problems forming clots spontaneously. Is that good? No, because then you have clots in your blood vessels. And sometimes those clots break free. So there's two different clots that you can have in your body. Thrombus, which is staying put, and what? The floaters. They're called embolus. And those are bad because you never know where they're going to end up. They could end up in your brain. They could end up in your lungs. They could end up in your heart. And if they block a pathway, everybody past it isn't getting dinner tonight. Yes? This is an example of an electron micrograph scan of what a clot looks like. So stuff gets caught up in it, and that's why we see the red blood cells, because it gets really sticky like a spider web. So some of the cells that go by might get stuck in the clot as well. Once we've repaired the damage, we can't just pull that clot off and let it go free, right? Because that could cause a problem. So they talk in your textbook about clot retraction and repair. So basically I'm going to get those fibrin solids dissolved by a process called fibrinolysis. And there's a couple of different chemicals that are going to help do that. Get the fibrin back into liquid form after the damage is repaired. Plasminogen and tissue plasminogen activator are some of the chemicals that will help dissolve that fibrin clot or fibrin mesh. Disorders in homeostasis of the clotting process. And again, hemostasis is what clotting is all about. Know the clotting cascade, yes? Only the part I showed you at the bottom of the diagram. And understand that clotting factors, even though you don't have to memorize them, are involved in the clotting process. So they define thrombus and embolus for us. That's a problem. They talk about after surgery and disruption of any blood vessels, you increase your chances of forming spontaneous clots within your circulatory system. So some disorders where the disruption in the circulatory system might happen, they give patients anticoagulants to help decrease the chance that they form these clots. They give us the oral version, which is called Coumadin, of an anticoagulant, or warfarin. And there's a IV given anticoagulant that they mention there as well. It's heparin. They both kind of do the same thing through different mechanisms. So depending on what the patient has or the condition the patient has, we might be able to give oral, or we might have to give intravenous anticoagulants for a while. They talk about disseminated intravascular coagulation, where we can spontaneously sometimes create these clots within our cells, and that's a problem. Bleeding disorders, when I can't make platelets, or I don't make them right, or I don't make enough, I'm going to have problems with this whole clotting cascade as well, because they're kind of the initiators of all this chemistry. So they talk about that as well. Thrombocytopenia, what's that? Not enough platelets to be efficient. Impaired liver function, again, is going to be a big problem. One of the very important vitamins you need for the clotting cascade is vitamin K. That was on your quiz. And then they talk about the factor that is usually deficient in hemophilias. Factor what? 8. Okay? And that's different. Different types of hemophilias may have different deficiencies. Remember when you donated your blood, you had that wonderful blood plasma? They don't just throw that crap away. Guess what? They can actually extract some of these specific factors. so they can use them for patients who have hemophilia as well. Where do we find these factors? In your blood plasma. So your blood plasma can be used for many different things, and many times we extract these factors out for different patients' needs. Blood transfusion and replacement, we already did the blood thing. Everybody good with the blood thing? How do I tell what blood type I have? That's how I'm going to end it up. Well, I go and I give my blood, and then they do this special test, and they tell me what kind of blood I have. Well, I can actually make antibodies to an antigen. All I have to do is take the antigen and put it in something that it's foreign to, right? So if I take my red blood cells that have the A antigen on it, and I stick it in a sheep or a goat or a bunny rabbit, those are the common ones we pick on, what are they going to do? They're going to make antibodies to that antigen. And all I have to do is draw their blood, take their plasma, separate the antibodies, and then I can put it in a little tube with a dropper and sell it to you in the clinical lab. You can then use those antibodies to figure out what blood type I have. So if I have type AB blood and I put anti-A antibodies from the sheep I picked on it, what's it going to do? It's going to clump. If I put anti-B antibodies on it, it's going to clump. That would be type AB blood. Okay? So my blood goes on there, and that packaged prepared antibody goes on there. If there's a reaction, it's there. Yes? If I have type A blood and I put my anti-A antibodies, it's going to clump. But what if I put the anti-B antibodies on there? Anything going to happen? No, because I don't have what? I don't have anti-B, I don't have B antigens on the surface of my cells, do I? No reaction. If I have type B blood, yes? Anti-A, nothing. There's no A antigens there. But what's going to happen with B? Yeah, anti-B is going to react with my B antigens, big clumps. And that's the size of the clumps that would be in your circulatory system. That's why that's a problem. How about type O? Nothing. Oh, yeah. Disseminated intravascular coagulation. Big time. Okay? And you'll see when we talk about immune responses, we will talk about why that happens. Okay? So that's a blood typing. So that's what they do. And you're going to do that if you want to in lab too. At least in my labs anyway. Okay? So if you put a little drop on a slide and I have my package prepare antibody, if there's a coagulation that occurs, you've got that antigen. Okay? So they talk about blood typing, transfusion reactions. Again, you'll have a reaction if you have, for some reason, been given an antibody to the antigens that you have, or for some reason you are given an antigen to which you have antibodies.
My question is, what if the person who's given the wrong type of blood transfusion already have those signs because of other tissues?
That's why, and I explained this to you, before you get any blood, and it doesn't matter what it is, if it's the same blood type as you, it doesn't matter. What they're going to do in the lab is they're going to do something called a type, figure out what type you are, and a cross match. So the first test they do is this. They figure out what blood type you are. Then they go into the fridge and they grab one of the bags, same blood type. And then they take some of the blood. And now they're going to put it in a tube and mix it. So they're going to put the plasma from the patient that's donating on your cells. And then your plasma on the cells from the bag that's donating. And they're going to mix them together. And if they clot, what's going to happen? They're not going to use that bag. Because it's not just A, B, O antibody-antigen reactions that we can see. Sometimes patients, for some reason, whatever it may be, may make other antibodies to antigens that might be on the donor's cells. Hopefully that answers your question. Huh? Oh yeah, there's a whole panel. There's huge, yeah. Yeah. Not just ABO by any means, but there's a whole bunch of other antibodies and antigens that can result from that as well. So they always do the type and cross-match before you get any unit of blood. Yes? It's in the, you know when you give blood, there's a big long tube and they crimp it? That's like a tube of blood. So now what's left in the tube, not talking about the bag, in the tube you have plasma and cells. And they crimp it all the way up. So you have probably about that much all the way, and they can take a piece, and it would be like having a tube of the donor's blood. Okay? Yeah, in the bag's washed red cells, but in the crimped tube, you have all the components. Yes? Are we good? Whew. You have a lot of studying to do. Okay? Especially on that hormones